Please proceed to take a seat. It's my great honor to introduce the second keynote speaker for today, Dr. Juan Helen Zhou. Helen is a professor at the Center of Sleep and Cognition and the deputy director at the Center of Translational Magnetic Resonance Research at the National University of Singapore. She received her PhD from Nanyang University in Singapore and then did multiple postdoc positions in the US, at UCSF, the Singapore-MIT Alliance, and NYU, before then joining the Duke NUS Medical School as a faculty member. She is also very active at the OHBM, having served at multiple positions here. And I don't want to take away a lot of her time, so I'm just want to mention that her work is really beautifully resembling and exemplifying OHBM with this mix between understanding brain organization and the basis of brain disorders, neurological and psychiatric disorders. And with that, please welcome Professor Helen Zhou to the stage. Helen.
Thank you so much, Simon. A very good afternoon, everyone. I'm Helen Zhou from National University of Singapore. I would like to first thank OHBM leadership for the kind invitation. It is my greatest honor to be here. My first OHBM was in 2006 in Florence, and since then, it has been my favorite conference. It is lovely to see all friends and colleagues after 2 years again. In the next 40 minutes, I would like to share with you our work on differential brain network phenotypes in neurological disorders from a longitudinal perspective. As you all know, given the aging society worldwide, the number of people with dementia will double every 20 years. Dementia is a family of neurodegenerative diseases that lead to severe cognitive decline, behavioral problems, loss of daily function. Alzheimer's disease, in short AD, is the most common type of dementia. 10 years ago, Jack Sperling and colleagues proposed a hypothetical model of the major AD biomarkers. It starts with extracellular amyloid beta plaques, and then intracellular neurofibrillary tau tangles, followed by brain atrophy, even at cognitively normal stage. These pathophysiological abnormalities precede memory decline and clinical symptoms by many years to decades. Given this model and the failure of multiple drug trials targeting late stage, the long-term goal is to develop individualized early detection and prognosis to facilitate proactive intervention. In other words, we hope to predict disease vulnerability and progression from the examination of an individual years before the appearance of any symptoms. To achieve this goal, today I will first introduce network-based neurodegeneration hypothesis using network-sensitive imaging methods. Under this framework, I will then focus on 3 key questions in the context of neurodegenerative and cerebrovascular disorders. Can we detect symptom-specific network changes in dementia subtypes? Can we track the longitudinal changes in pre-dementia stage? And lastly, review disease mechanism and predict progression. To start, here are the 5 T1-weighted structural MRI of patients with 5 different neurodegenerative syndromes. As you can see, all 5 disorders have very distinct atrophy patterns. For example, AD patients usually lose their episodic memory and visual spatial function, causing atrophy in a posterior brain network involving posterior parietal, posterior cingulate cortex, hippocampal, and temporal regions. In contrast, behavioral variant frontotemporal dementia, in short BVFTD, disrupt the social emotional functions and target an anterior network, including frontal insula, anterior cingulate cortex, and subcortical regions. Based on evidence from neuropathology, imaging, and animal models, neurodegenerative diseases have been thought to relate to a network-based dysfunction. Each disorder targets a specific large-scale neural network. In 2009, Bill Seeley led a team to test this hypothesis in living humans for the first time. We used network-sensitive imaging methods, which include resting-state fMRI and structural MRI. Resting-state functional networks, or intrinsic connectivity networks, map temporally synchronous, spatially distributed, spontaneous, low-frequency both signal fluctuations from four-dimensional fMRI data. Besides low-level somatomotor and visual networks, high-level cognitive networks such as the default mode network, attention networks, executive control networks, and salience networks can also be extracted. So starting with the atrophy pattern of each disease in the cyan color here, we pick the most atrophied region as circled here by performing seed-based network mapping using resting-state fMRI data of healthy participants. The resulting functional networks are highlighted in yellow. You can see great similarity between functional networks in health and atrophy patterns in disease. Similarly, using the same set of seeds, the structural covariance network derived from structural MRI of healthy controls is shown in green. It spatially resembled the other two maps. There is a direct link between functional networks and gray matter structure in health. So taken together, we have confirmed the network-based neurodegeneration hypothesis in living humans. Regions wired together, they found together, grow and shrink together. When the disease starts, regions within the same target network also start to have atrophy together. This work leads to many interesting questions on selective network vulnerability along disease progression. Following the previous model, several groups refined the hypothetical model of AD by including resting state network connectivity as the possible marker. Let me start with the first question. Can we detect symptom-specific network changes in dementia subtypes? AD targets the deformed network, in short DMN specifically. This is supported by increased amyloid burden, atrophy, and hypometabolism in the same network. In 2004, Mike Gracious from Stanford performed this pioneering work, which found that AD patients have reduced default mode network connectivity compared to control, which is independent of atrophy. How about the other dementia subtypes? A few years later, we have shown that AD and BV-FTD featured divergent connectivity changes in the default mode and salience network, corresponding well with the opposing symptom deficit profiles. BV-FTD had reduction in the salience network, but increased the connectivity in the DMN. In contrast, AD had reduced default mode network connectivity, but increased salience network connectivity. So this finding supports the network-based dysfunction hypothesis and has been replicated in quite a number of studies. One step further, besides the within-network connectivity, we hypothesized that inter-network connectivity would be differentially disrupted in these two disorders, underlying their cognitive and social-emotional deficits. To test this hypothesis, our team decided to systematically study whole brain functional connectivity in AD and BV-FTD based on a Singaporean cohort. At the whole brain level, you can see both disorders had reconfigurations in functional network structure compared to controls. When we narrowed down to specific networks, AD had more fragmented modular structure in the default and control networks, while BV-FTD had more severe damage in the salience network and subcortical regions. Importantly, such connectivity profiles track the disease severity and clinical symptoms. Across BV-FTD patients, lower salience network connectivity in the right frontal insula was associated with more severe disease severity. Now, by considering inter-network connectivity, we found that increased salience network participation coefficient, indicating loss of modular structure in the x-axis, was related to more severe behavioral problems across all patients. Within the same disease, such as AD, there could be several syndromic variants supported by different network patterns. Several works have tried to explain the clinical variability in AD using resting state fMRI. Neyman and colleagues found that syndrome-specific patterns in 3 AD variants, as shown here, were driven by the involvement of specific networks outside the core default mode network. For example, the early onset AD was associated with anterior salience and right executive control network, supporting their profile of more attention and executive dysfunction, but relatively spared memory. On the other hand, AD frequently coexists with cerebrovascular disease, in short, CVD. CVD is associated with declines in executive function and processing speed. AD with CVD is the most prevalent dementia subtype, especially in Asia. CVD MR markers typically include lacunes, white matter hyperintensity, microbleeds, and so on. CVD and AD have been proposed to have additive effects on cognitive decline. So for the same dementia severity, AD-only patients present with more severe AD pathology than those comorbid with CVD. Based on this, Joanna Chong from my lab asked this question: would we see a similar or differential network patterns in AD with and without CVD? Based on a memory clinical cohort from Singapore, for the first time, we showed that AD patients with and without CVD featured divergent functional connectivity patterns in the default mode network and executive control network. By comparing the CVD and non-CVD groups with the same disease severity head-to-head, AD without CVD had more reduction in the default mode network connectivity. Lower DMN connectivity was related to lower hippocampal volume. In contrast, CVD group had higher frontal executive control network connectivity, which is correlated with greater burden of white matter hyperintensity. Interestingly, these functional network changes were also spatially mirrored by the structural covariance networks. And this work supports differential network phenotype in AD with and without CVD, which reflect underlying pathology. Besides functional network breakdown, white matter degeneration is also an early event in AD, which closely follows the A-beta deposition. Diffusion MRI measures the movement of water molecules. It is often used to examine white matter microstructure and structural connectivity. However, the traditional single tensor diffusion model suffers from partial volume effect. In other words, the signal in the voxel is a mixture of tissue compartment and water content. How can we solve this problem? Dr. Postulak from Harvard proposed a biotensor free water method. It separates the signal of each voxel into 2 compartments. Tissue compartments represents exon bundles, and free water compartment represents extracellular water. Changes in tissue compartments, such as reduced fractional anisotropy, are related to exon damage and demyelination. Free water increases could be caused by vascular damage or neuroinflammation, possibly due to blood-brain barrier leakage. In collaboration with author Ji Fang in my lab is interested in the differential white matter changes in AD with and without CVD. Compared to the original diffusion tensor imaging results shown in the first row, the free water corrected tissue compartment at the bottom row revealed more focal microstructure differences in the frontal occipital parietal regions between the two groups. The contamination from the ventricles was minimized. When we look at the free water, it is not surprising that AD with CVD and VAD had widespread higher free water in normal appearing white matter. But interestingly, you notice that AD without CVD group did not differ from control in terms of white matter hyperintensity. But AD without CVD also have higher free water in normal appearing white matter, suggesting inflammation or microvascular changes. Higher free water in normal appearing white matter relate to dementia severity across all patients, while the white matter hyperintensity did not show this relationship. Building upon this previous work and the increasing awareness to the brain-heart connection, we hypothesized that higher brain free water would be related to cardiovascular dysfunction measured by circulating blood biomarkers. And brain free water would mediate associations between cardiovascular biomarkers and cognition. We test this hypothesis in more than 300 older adults from a memory clinic. We found that higher cardiovascular biomarkers, such as high sensitivity troponin T, was indeed related to higher brain free water in widespread white matter regions and specific gray matter networks, including default, control, attention, and low-level networks. Importantly, free water increases in both gray matter and white matter mediate associations of baseline cardiovascular blood biomarkers with longitudinal decline over 5 years. So the pattern for white matter is widespread and applies to both memory and executive function. However, the pattern, in contrast, the gray matter free water exhibits this network and domain-specific pattern. Higher free water in the executive control network mediate the relationship with the executive function, but the deformal network specifically are responsible for the memory. So this study provides new evidence for brain-heart connection, highlighting the role of brain free water increases in linking cardiovascular dysfunction to cognitive decline. Taken together, these findings based on structure and diffusion and functional MRI lend further support to network-based dysfunction hypothesis at dementia stage. Sorry, I forgot to take off my mask. Brain network phenotypes explain clinical variability, track symptom severity, and relate to AD and vascular burden. So now, let me move on to the second question, tracking longitudinal brain network changes in pre-dementia stage, which is more critical for early detection and intervention. Building upon our cross-sectional study related to AD and CVD described earlier, together with Dr. Zhou from South Korea, we are interested in whether amyloid and cerebrovascular burden are associated with divergent longitudinal trajectories of functional connectivity in mild cognitive impairment, which is the prodrome stage of dementia. And this time, we have direct objective measures of amyloid burden via PET and longitudinal resting state fMRI data. Again, we focus on the default mode and executive control network connectivity. To test the hypothesis, we directly compare the two pure subtypes at a prodromal stage, amnestic MCI PIP positive and subcortical vascular MCI PIP negative. Prodromal patients with pure amyloid burden, amnestic MCI PIP positive, showed steeper declines in the deformal network connectivity represented by red lines here. In contrast, prodromal patients with pure cerebrovascular burden, subcortical vascular MCI PIP negative, showed steeper increases in executive control network connectivity represented by green lines. If we include the other two groups with mixed pathology and repeat the analysis in a continuous manner, we found the same thing. This finding replicate previous study and highlight the divergent influence of amyloid and CVD pathology on brain functional network trajectory in pre-dementia stage. Today, most functional cognitive studies, including ours, have typically quantified CVD burden using a single MR marker, such as Lacun's or white matter hyperintensity. However, we know that CVD comprises multiple MR markers, such as microinfarcts, microbleeds, etc. The etiology and disease severity of CVD markers are heterogeneous. So it is unknown whether different markers exert differential effects on functional connectivity. So very recently, Joanna Cho from our lab employed a multivariate approach taking into account 5 different CVD MR markers. We found widespread, non-marker specific effect of CVD pathology on brain functional connectivity. Higher CVD pathology was generally linked to lower cortical intra-network specialization, except the control network, and lower inter-network segregation. It is also related to lower subcortical functional connectivity to transmodal networks and higher subcortical connectivity to unimodal networks. Importantly, the greater degree of such general CVD-based network phenotype was linked to lower visual motor function. You're welcome to drop by our poster to know more details from Joanna. So that is the functional network. What is the influence of amyloid and CVD burden on white matter structure in pre-dementia stage? Using free water imaging on adenine data, Ashwati from my lab found that A-beta positive cognitive normal elderly, represented by red lines here, showed a greater reduction in fractional anisotropy in the right arsenic fasciculus over time. This suggests that A-beta deposition is related to early white matter degeneration. In terms of CVD burden, Sama and Siwei Liu studied the relationship between a wide range of CVD markers, multiple cognitive domains, and white matter microstructure in non-demented elderly population in Singapore. Using path analysis, white matter abnormality mediated the relationship between CVD and cognition. Specifically, Lacuna-Kant's white matter hyperintensity volume and intracranial stenosis were associated with executive and verbal memory impairments via white matter disruption in commissural fibers. In contrast, the impairment in the visual memory and attention and language were mediated through projection fibers. On top of these standard CVD markers, in collaboration with Leiden and colleagues, Li Wen from our lab is interested in a novel CVD marker named corticocerebral microinfarcts, in short CMI. It often appears as the little line perpendicular to the gray matter surface in FLAIR or T1 images. CMI is more prevalent in AD and related to cognitive deficits. However, the underlying mechanism remains unclear. Animal models of CMI suggest that it may induce white matter impairment, neuronal loss, and decreased response in remote cortical areas. Therefore, we hypothesize that CMI may affect cognition by disrupting the local and long-range white matter pathways in living humans. Indeed, using fiber tracking, CMI led to lower structural connectivity efficiency and degree centrality, specifically in the executive control and dorsal attention networks, in prodrome stage only. So the cognitive impairment, no dementia, in short CIND here is the prodrome stage, and such effect was missing in dementia stage. The structural network breakdown mediated the association between CMI and cognition. This confirmed for the first time that CMI influenced the cognition via the structural network breakdown involving remote regions, especially in pre-dementia stage. Besides cognition, similar stage-dependent effect was also found when we examined the associations of brain with neuropsychiatric disorders. So in individuals with CIND, So the higher total neuropsychiatric inventory severity in short MPI score was associated with widespread free water increases in white matter, as shown in red here, and focal tissue fractional anisotropy reductions in frontal parietal and callosal fibers in blue. So in addition, as you can see in the plots at the bottom, prodrome patients with more severe white matter degeneration at baseline also had a faster increase in the MPI score over time. But in contrast, in dementia stage, we did not see such pattern in white matter. Instead, only the cortical thinning was related to higher total MPI score at baseline, and it was not predictive of future MPI changes. So again, this highlights the important role of white matter degeneration in early stage of the disease. Now, let us talk about the very early preclinical stage. The key question here is whether the longitudinal network changes would reflect the cognitive decline over time in cognitively normal elderly, and what is the potential influence of risk factors such as ApoE genotype. We know that the human brain is functionally organized in a highly modular and distinct manner, with high connectivity within network indicating functional specialization and no connectivity between networks representing functional segregation. Work by Chen, Bazel, Spray, and others have demonstrated this age-related loss of functional segregation and specialization cross-sectionally. The age-related between network changes is especially prominent in higher order cognitive networks. For example, the task-positive networks and task-negative networks are often anti-correlated or segregated. Mellon and Udin proposed the triple network model, which hypothesized that the primary role of the salience network, which is connected to subcortical regions, is to enable the switching between two states of the brain, the default mode internally guided state and the task-related externally related states. So we hypothesized that the longitudinal trajectory of network specialization and segregation in these higher order cognitive networks would reflect longitudinal cognitive decline in aging. Eric Ng and Ingwei from our lab studied these longitudinal changes in connectivity and cognition over 4 years in a group of Singaporean cognitive normal elderly. We found longitudinal functional connectivity decline within the default and executive control networks over 4 years. Moreover, aging is associated with this age-dependent decline in functional segregation between the two networks. Interestingly, when we divide the participant into ApoE4 carriers and non-carriers, ApoE4 carriers who are at higher risk of developing dementia, denoted in orange line here, had faster loss of functional segregation over time, suggesting an accelerated aging pattern. Greater loss of functional segregation between these two networks on the x-axis was also associated with faster decline in processing speed over time. Besides processing speed, decline in cognitive flexibility is also a prominent feature of aging. It often results in an inability to adapt to new situations and environments. A few years ago, a study from UPenn reported that the degree of alignment or liberality between structural connections via diffusion MRI and functional brain signals during a cognitive switching task was associated with cognitive flexibility in young adults. Higher function structural liberality was related to worse cognitive flexibility. Wan Ning from my group applied the same method in a longitudinal manner, and this time we used resting state fMRI of healthy older adults instead. We found that salience network have reduced function structure alignment and increased liberality over time, while the deformal network showed the opposing trend in aging. Older adults with greater decrease in function structure alignment and increase in liberality of the salience network had faster decline in cognitive flexibility over time. This finding suggests that aging differentially impacts the extent of structural constraint on functional signals in a network-specific manner, even without any task. It also underscores the critical role of the salience network in cognitive flexibility, which supports the triple network model. Another observation is that most previous studies are based on assumption that the brain and behavior relationships are constant across all stages. However, as you can see, the hypothetical AD model basically tells us that the different biomarkers are following a nonlinear trajectory from preclinical to dementia stage. And there might be interactions between multiple factors and pathways, and how to handle this problem. Multivariant approaches will be very useful, which can be applied to not only structural MRI data, but also PET data. Here's one example. Under the framework of network breakdown, we ask the question whether and how the influence of network-based neurodegeneration on cognitive function vary with disease stages and pathology. To answer this, Qianxin from my lab derived the individual-level metabolic network scores from FDG PET and structural covariance network scores from structural MRI based on more than 700 adenine participants using partial least square approach. As you can see, both networks nicely resemble the key canonical brain networks. We then used a sparse varying coefficient model, which does not assume a constant relationship between brain network measures and the memory score across stages. It also allows the selection of significant predictors with lasso. So overall, we found that the association of structure and metabolic networks with memory exhibit these pathology-dependent nonlinear trajectories across stages. Specifically, in amino-positive group on the left, the association between hippocampal structure network and memory was strongest in cognitive normal stage. It then gradually decreased with disease progression. This suggests an early influence of hippocampal structure network deterioration on memory impairment in preclinical AD. In contrast, such hippocampal memory association was overall weaker in non-AD groups, and it has a very different trajectory, as you can see on the right-hand side. In terms of metabolic network highlighted in yellow, in the red color here, we observe a biphasic role of the angular gyro-seated deformal network in memory only in amino-positive group. The influence is higher in both preclinical and dementia stages. So our finding provides the first evidence for pathology and stage-dependent influence of metabolic and structure networks on memory, which support AD biomarker model. Till now, we have learned the longitudinal trajectories of brain network changes in pre-dementia stage, the influence of genetic risk factors, different pathology, as well as their stage-dependent relationship with cognitive and behavioral impairment. Lastly, we hope to build up disease progression predictive models and understand mechanism. One key question is whether brain connectome can give us insight into mechanism of network breakdown. We have demonstrated this possibility at the group level by comparing healthy connectivity graph to atrophy severity in disease. For example, based on the healthy functional connectome, region having shorter functional paths to the AD epicenters and higher connectivity flow to other regions within the target deformal network also had greater atrophy in patients with AD. Such association between healthy connectome and atrophy severity in disease has been validated across the 5 dementia subtypes, which fit best with the transneural spread model and to a lesser extent, nodal stress model. The transneural spread model proposed that misfolded protein propagate throughout local and long-range circuits via transsynaptic spread, which then lead to network breakdown. The nodal stress model or anterior model suggests that the key synaptic convergent zones might be more vulnerable and disconnect functional circuits. However, these group-level findings are just the first step towards connectome-based prediction of disease progression. In 2019, Jesse Brown extended this framework to the individual level. They show that rather than group-level disease epicenter, unique atrophy epicenters can be detected in individual patients with FTD. Then we can use these patient-tailored epicenters in a connectivity-based model to accurately predict individual longitudinal spread of atrophy. And this finding, again, supports the transneural degeneration hypothesis. Inspired by this study, we realized that such connectome-based prediction effort has not been done in AD. So one step further, we aim to build up a precise and interpretable AD progression model to forecast future brain nodal atrophy and cognitive decline at the individual level using multimodal brain imaging data. We decided to start with functional connectome from resting state fMRI and leverage on-graph convolutional network, which incorporates both brain network architecture and nodal features. Based on two independent data sets, ADNI and a Singaporean data set, we demonstrate that the functional connectome-based deep learning model can successfully predict regional atrophy progression at the individual level. And this is done for the next 3 years across different stages of the disease. Moreover, we identify the network-specific contributions to such atrophy progression prediction. As expected, the targeted network connectivity contributes most to its own atrophy prediction. And importantly, hippocampus, default, and control network connectivity at baseline were the top 3 predictors for all networks' atrophy prediction. We have also demonstrated the feasibility to use this model to predict future cognitive decline and dementia severity progression. And feel free to interact with Xiaoyu during oral and poster sessions. The connectome-based prediction models apply to not only atrophy, but also amyloid and tau deposition. It can also be derived from diffusion MRI. For example, this figure demonstrates that the anatomical distance to outbreak regions modulated the amyloid propagation process. Regions closer to the disease epicenter also have earlier amyloid arriving time. One step further, one very recent paper in Neuron studied the interactions between A-beta and tau, the trigger and bullet in AD. They identify 2 stages in the onset and acceleration of AD tau spreading. Stage 1 is driven by the brain connections to entorhinal cortex, which features the spreading of tau from entorhinal cortex to its connected temporal and limbic regions. Stage 2 consists of tau propagation into the neural cortex, which is dominated by connections to inferior temporal gyrus. This highlights the complexity of network-based spreading model, and at the same time paved the way for identifying critical window for AD therapeutic strategies, such as those mentioned by Professor Lozano yesterday. We need to take note that such observation of unifocal connection-based spread is only one of the possible network breakdown scenarios. It does not take into account genetic vulnerability and other biological factors. It can be staggered multifocal onset, which is not related to connectome at all, or it could be a combination of the two. More work is needed to understand onset and progression of multiple disease pathologies and their interactions. In conclusion, I hope I've convinced you that it is important to examine longitudinal brain structure and functional network breakdown trajectories in neurological disorders. Network phenotypes link to symptoms and pathology, clinical variability, and disease mechanism. It also has the potential to predict disease progression. Moving forward, to achieve our goal of individualized early detection, prognosis, and proactive intervention, we need to leverage on multimodal imaging, digital tracking, blood markers, and genetic assays. Longitudinal network sensitive imaging methods, combined with computational approaches, with high reliability, reproducibility, and interpretability, as Sarah mentioned perfectly this morning, will allow us to systematically test the predictions made by network breakdown models. This one then helps us to tackle the heterogeneity and improve stratification for intervention. I'm currently part of a 2-year clinical trial called SINGER in Singapore to evaluate the efficacy of multi-domain lifestyle intervention to reduce vascular cognitive impairment. The key question we ask is whether brain measures and other biomarkers are predictive of intervention response at the individual level. And I hope to report to you guys in a few years. So with that, I'd like to thank the funding agencies, all my collaborators and lab members contributed to these studies. You're welcome to drop by our posters highlighted here, physically or virtually. And lastly, our center is actively recruiting for faculty, postdoc, and PhD students. And please check our websites here. Welcome to email me if you're interested in these opportunities or any collaborations. And thank you for your attention. I'm happy to take any questions.
Thank you very much, Dr. Zhou. That was an excellent talk. And there are some questions from the audience. So some of the people in the audience were wondering if you could elaborate a little bit more about how the free water was estimated.
So the free water is estimated by this biotensor model. So basically, you model a sphere on top of the single tensor model and try to estimate the free water measures from that sphere. So it is very similar to the measure of mean diffusivity, but it's more specific to the extracellular free water. And the optimization algorithm involves some iterations for sure. And in order to estimate free water, nowadays you probably need to have multi-share diffusion MR data.
Okay, also another question that became popular was the question of identifying individual subjects. So you hinted towards the end of using this on individual subjects, and of course you talked about longitudinal studies, but what do you think the sensitivity is for looking at this in individual patients?
Identify
individual
prediction.
Yes.
Okay, the sensitivity part, right? Yeah. So I think we're not there yet. Because you can see the last study we demonstrated the R value is around 0.45 in terms of predicting the nodal atrophy, right? So that's at the individual level. But when we try to predict their future cognitive decline and dementia severity, the R value approached 0.708. Right. So, however, you can see this is only based on resting state fMRI data. We haven't included the other modalities yet. So right now, our team is trying to integrate diffusion MRI and other modalities and see whether we can increase this accuracy further. And obviously, the interpretation will be also slightly different. So I think we need more work. Yeah.
Okay, and another question is whether we can differentiate the cerebrovascular pathology and cognitive decline that is part of typical aging from beginning dementia.
Okay, so this is a good question. Right now, I think in the field of CVD, people make some diagnosis of cerebrovascular disorders based on those CVD MR markers. And then there are certain kind of arbitrary there in terms of, okay, there are white matter hyperintensity rating greater than something, or it is CVD, and lower than something, it is not CVD. So I would rather prefer a more continuous manner to analyze that data, and obviously that will take into account the whole spectrum of the individuals with microvascular damage. That's the whole point of looking at free water, actually, because I emphasize that even your white matter looks normal from the scans, your free water in those normal appearing white matter is already increased, and it's actually predictive of future dementia severity, CDR sum boxes, and cognitive decline. And then how to differentiate from the normal aging, that is a really hard question. I feel like we have to really have all the comprehensive biomarkers in terms of all the, pathology in order to answer that. But right now, the data is limited. Yeah. So I feel like the CVD pathology or cardiovascular dysfunctions and all the risk factors related to CVD is really probably part of the natural aging processes, So in our analysis, you could actually use multivariant approaches to control for all that and try to isolate some latent factor, hoping that, one latent factor is going to be responsible for AD, another is responsible for CVD, and another is for normal aging. So this, we wait and see.
Okay. A few other questions are coming up here, a number of questions, obviously. I'm going to pick one of the top ones. How do baseline perfusion or CBF measures, how do they change or affect the functional connectivity measures? And a related question that I have of my own is that usually you see a global hypometabolism. Did you see, even in the network structures that increase connectivity, was there a hypometabolism as well? But so, kind of a two-part question. How does the baseline perfusion CBF affect the changes, the connectivity measures?
So for the first question, how the baseline diffusion or structural connectivity influence the functional connectivity, we actually didn't test it directly. Let me think whether we have done something like that. We always do it in parallel. We didn't really test them directly. And our lab is also kind of interested in the structural functional relationships. That's why we are using this method by Daniel Bassett in terms of the structural functional alignment and liberality. And people also try to do the correlation between structural connectivity and functional connectivity. So what I can tell right now is really they influence each other, right? So structural connectivity can lead to increased functional connectivity. If there's a remyelination, then functional connectivity will increase. But the functional connectivity or synaptic interaction can also bring, or disconnectivity can also bring damage to the structural connections. So it's a bidirectional processes. Yeah. Second question is related to
just
hypometabolism.
Sort of my own question of usually you see maps of hypometabolism, like they're kind of global, but it seems like some of the networks actually increase connectivity that were overlapping with the hypometabolism. It seems like some of the areas of hypometabolism that you typically see were in nodes that showed increased connectivity with Alzheimer's. So.
Oh, I see. Actually, I won't say that hypometabolism is global. I would say it is quite network specific as what we see. Okay. Usually, hypometabolism in the field of AD is considered one marker of neurodegeneration. Yeah. If you're thinking about increased functional connectivity, that might be related to the preclinical or pre-dementia stage. There's always a U-shaped trajectory that is pretty common. Yeah, and whether that is compensatory or aberrant, that requires a more study. Yeah, so these are two competing hypotheses. Yeah.
All right, well, thank you very much, and thanks for the great questions, and thank you for the answers. Thanks.
Thank you.